What's good, y'all, and welcome back to another edition of Mr. Fitzgerald's biology class. Because it's been a while since we talked about some of these topics, we're going to run through them in preparation for our upcoming exams. So I wanted to start with Unit 1 today, talking about water chemistry as well as the macromolecules, which we had done in the past with Unit 2, but I'm going to bring them into Unit 1 for this. Now, I'm going to go fast through a lot of these topics and try to explain some things. If you need to, pause the video so you can process the information, but we're going to get rolling. So we're going to start off with water because it was one of the key things within Unit 1, and it has a lot of vocabulary with it. So let's look at some of the water vocabulary. Before we do that, let's think about the structure of water. Remember, water is made up of oxygens and hydrogens. You have two hydrogen atoms and one oxygen atom here, and you have positives and negative ends. The hydrogen atoms have a slight positive charge to them because their electrons are actually being pulled away by the oxygen. So that way oxygen has a slight negative charge. Because oxygen is gaining a little bit more electron presence by pulling those hydrogen's electrons towards it, it gets a little bit more of a negative charge because electrons are negative. So what this allows to happen is hydrogen bonding, these interactions between molecules. So this water molecule up here with its hydrogen that's slightly positive and its slightly negative oxygen is going to align itself so that way the positive on this hydrogen can be next to the negative of this adjacent water molecule's oxygen. And those positive ends will be pointed away from the positive hydrogen. And the whole concept here is that opposites attract. So you have the positive hydrogen trying to attract to that negative oxygen and pulling each other. So that way there can be some electrical charge balance overall. Now, what this allows is things like cohesion to happen or sticking together. So sometimes we also call this surface tension. If you have an object that's floating on top of water, the reason it's floating is because water molecules are literally holding hands and attracting to each other, right, through those hydrogen bonds that we saw on the previous slide, and it's preventing the object from kind of descending downwards. So for instance, imagine all the water molecules together at the surface of the water kind of acting like a large net, so things like boats or other objects don't just sink right through, they kind of get supported by the net overall. So that's a cohesive process. Adhesion is when water will stick to a surface. So because water has an attractive quantity to it in itself, with cohesion, it can also stick to surfaces as well. This is why when you have a glass of water and you pour it out, you still get some droplets in there. They're trying to hold on to that surface as well. All right. This also allows something called capillary action, which is kind of a cool process, and it's anytime that you have a straw inside of a restaurant and you have a little bit of liquid in the cup, this liquid in the straw always is a little bit higher than the cup because most of the time that liquid contains water, and water will actually move against gravity to crawl up that tube, basically trying to spread itself out. So it does give the water the ability to flow upward, and this allows plants to get water from the roots where they absorb it all the way up the stems and all the way to their leaves and other parts of the plant because it has these thin tubes in it that actually allow water to basically move itself. It doesn't have to actually use energy to have water flow. All right. So more water vocab, fast and furious. That water is the universal solvent. Remember, a solvent is anything that can dissolve something. And water is very good at dissolving many things, like salts, like sugars. It can mix with a lot of things overall. So we give it this universal solvent idea. There are some objects that water does not mix well with. Usually lipids, fats, oils, waxes. It does not like water. Those are hydrophobic things. Things like salt and sugar are hydrophilic, so they'll dissolve very well in water. All right. Another cool concept of water is thermal expansion, and this allows ice to float. And what basically happens here as water condenses down to a freezing state, instead of its molecules condensing down and getting tight together, they actually spread each other apart. And the reason that they spread apart as they lose energy and kind of become a little bit more motionless and vibrate in place is that they have those positives and negative ends. So the positive ends of the oxygen want to kind of the positive ends of the hydrogen want to stay away from the other positive ends of the adjacent hydrogens, and the negative oxygens want to stay away from each other as well. So all the molecules, they can't spin away from each other like they normally do. So as they freeze, they kind of get in this perfect grid-like structure, which causes more surface to be between them. Liquid water has a little bit more motion, so those things can kind of flip-flop between. All right, but this overall keeps organisms alive. So when freezing temperatures happen, usually the surface of that water will freeze, and then the lower depths will actually be available for the organism to live in and are warmer. As you can see in this chart here, the surface freezes here at 0 degrees Celsius, and down below it's 4 degrees Celsius. It's not warm by any stretch for our imagination, but it's not freezing, so this fish will be able to survive down below. All right. Also, another cool thing with water is how fast and technically how slow it heats up. Water heats up very, very slowly. So this actually helps to regulate a lot of temperatures. This is why in coastal areas like here in Tampa, Florida, we don't usually see large temperature swings. Yes, we do get some days that start off really cold and get really warm, but overall, our kind of graph doesn't have a lot of spiking. And it's not even just for the day, for the whole time. So if you look at Tampa, or any coastal city, you usually see, it's moderate during the winter months and it's a little bit warmer in the summer. Whereas if you were to look in the middle of the country, right, and compare kind of the average temperatures of the month, you would have these very extreme lows and these very high highs. So they get larger swings overall. Yes, we do get temperature swings, but we don't on average go from -2 Celsius, which would be less than freezing, all the way into the 90s. So we don't get that. We usually stay somewhere between the 90s and the 60s. We'll have those days that get a little bit cooler, but we don't have a large temperature swing of 60 degrees. We only have a 30-degree swing in Florida. All right, so that was water. Now let's talk about chemistry. Some big things with chemistry are reactants and products with us. So remember, reactants are the things participating in the reaction. So these items that are coming together here, this methane and this oxygen that are coming together to make carbon dioxide and water here in this combustion reaction, these would be my reactants because they are being combined together to make my products. Products are on the right side of the chemical equation, or whatever the arrow points to. The arrow points to the products. Now, every reaction does require some sort of input of energy, and we call that the activation energy. To get anything started, you got to put in a little bit. So just think about it. Your car has a lot of energy while it's moving, right? You burn the gasoline to get the car moving, but to get it started, you have to turn the key, which kind of works as an ignition, sparking it, gets it going. So you need an initial spark, right? So basically, that activation energy is that minimum amount of energy to get it going. So going from our methane and our oxygen to get it started to be able to make our products of carbon dioxide and water. So that is that overall driving force within our chemical reactions. Now, what biology kind of incorporates into that is this idea of an enzyme, which is a type of protein that speeds up that activation energy by lowering it. So instead of having to go this large jump here and put in so much energy, if you have these biological catalysts called enzymes, these proteins, they're able to kind of reduce that amount of energy input. They orient the things, meaning they arrange them the way they need to be. They help line them up and they help reduce the energy and put them together. And then they break apart. So in this case, then we have something happen where it looks like it breaks apart because I have lower energy here. But this catalyst, this enzyme, is making it so much easier by lowering that activation energy, okay? Some big notes with enzymes is that enzymes are catalysts because they do speed up the reaction by lowering the activation energy. So the reaction does go faster, it does require less energy. But the key thing to note is not all catalysts are enzymes. There's things like iron and nickel and palladium and all this other stuff. Platinum, not palladium, that are different catalysts. They do speed up reactions, but they're not biological units. They're not proteins, okay? So anything that's a protein would be an enzyme here if it's a biological catalyst. All right, now that brings us to our macromolecules since we kind of talked about proteins. So remember, macromolecules are any big molecules in the cell, like this protein here. You notice there's a lot of chemical bonding in here. There's a lot going on. There's 4 different types. You got your carbohydrates, you got your lipids, you got your proteins, and you got your nucleic acids. Let's dive into each one individually here. So carbohydrates, remember those are sugars. Okay, when people are talking about carbs, they're talking about sugar. And sugar is a primary source of energy for our cells. It's a very easy, cheap resource that our cells can constantly use. Usually the elements that are involved in this are carbon, hydrogen, and oxygen. So C, H, and O are usually the things that are found in all sugars. And the big thing that you got to remember is anytime you see a hexagon on a page in biology class, that should just be a red light to you saying, hey, that's a sugar, okay? That's a type of carbohydrate. So I got carbons, hydrogens, oxygen, I got hexagon shapes, and These give me energy. Now, these hexagons can be kind of arranged differently into things called polysaccharides. So when you have long chains of these, like these starches over here, or glycogen that's in our muscle tissue, or even cellulose that makes up the outside of the cell wall for plant cells as well as bacterial cells, those are going to be polysaccharides, or many hexagons that are put together. Alrighty. Now, we got the lipids. Lipids are, again, an energy source. They're typically your fats, oils, and your waxes, and they're long chains of carbons and hydrogens. Okay, so anytime you have a large amount of carbon and hydrogen on something, and I'll show you a picture on the next slide here, that's going to be a lipid, and its main job is to store energy, because every single chemical bond that you see there, every single line that you'll see in this next picture represents a chemical bond, and when bonds break, they provide energy. So let's look at some types of lipids here. We have our saturated fats, and you can see all these lines here represent chemical bonds. So every one of those that you break, you get an energy release, as well as unsaturated fats. Remember, saturated fats are when they're filled. So if you look at all these carbons, imagine them as the table and hydrogens as the chairs at the table. Ignore this little part at the end. We're not going to pay attention to that. In this biology class, it's for AP Bio or some higher level. All right? So in here, you got basically all the chairs at the table are fit, right? You can't fit any more people at the table. The table's full. Over here, though, this table's kind of bent, right? And so you got, this side's pretty full right here, this side's pretty full right here, but you got a little bit of space right in here where you could probably fit a chair or two more, but there's no space for it, okay? So overall, this one has a little bit of gap in here. You got some areas where you could put some more hydrogens. This one's filled with hydrogens. So that's the difference between saturated fats and unsaturated fats. And those are two types of lipids. Another type of lipid that's important is the phospholipid. And these are the lipids with the hydrophilic head, the area that likes water at the top, and the hydrophobic tails, the areas that don't like water at the bottom. And these will make up the cell membrane. And remember, when they make up the cell membrane, they make it up in the phrase of a phospholipid bilayer. And what that means is that you have these hydrophilic heads face outside. So they'll either face inside the cell where there's a water environment, or outside the cell where there's also a water environment. And these tails will line up together in the middle. So basically, you've got these tails that don't like water in the middle, you got the heads that like water on the outside. And what this allows is what's called selective permeability. So I have substances that can't get through because these tails, right? So something like salt that has a positive charge or a negative charge on it. These tails hate charges. They're hydrophobic as well as nonpolar. So they'll push things away. So it keeps things from just going in and out of our cells whenever they want to. And it allows the other parts of diffusion and osmosis and stuff to work properly. So keeps our cells from getting overwhelmed by stuff. Now, a protein is a type of, is a macromolecule that's made up of a bunch of amino acids. And amino acids are things that are made up of carbons, hydrogens, oxygens, and nitrogens, okay? And as you know from units that we've already covered so far, there are 20 different types of amino acids, and we use our codon charts to figure out which amino acid is going with which which codon, which is a form of mRNA. Now, these amino acids overall have different shapes and sizes, but the main components is they all have this carbon, hydrogen, oxygen, and nitrogen that make them up. Now, what do proteins do? They're anything that do things really for the cell. Could be as simple as providing structure, being like some parts of the cytoskeleton or something like that. They could be used in transport. Okay, so any of those transport things, like those proteins that are either used in active transport or even facilitated diffusion, the ones that just provide doorways for things to move in the cells. They can be communication signals. Interferon's a great example of that. That's a protein that's produced by our cells when there's a viral infection to warn other cells that there's a virus within their area. It could also be something that speeds up a chemical reaction. So those would be your enzymes. And they control cell growth. So they regulate those cell growth and division processes. And when those don't work properly, then we have cancer or uncontrollable cell growth that could occur. So can we increase the speed of an enzyme? Of course we can. A couple easy ways to do it. Increase temperature. Think about it. Things that are hotter tend to move a little bit faster. Like next time you look at something cooking on the stove, there's a reason we heat it up to make it cook, because it gets everything moving a little bit faster. You get that boiling, that roaring action inside of your pot or whatever. You can increase the concentration of what's reacting. So if I want to get things to build faster, I can have more parts. Right? If I have more parts, it's easier for me to grab them and build stuff with them. Or I can have more people building things. I can have more concentration of enzyme. The more builders that I have available, the quicker the job gets done. So those are some easy ways to increase overall the speed of a chemical reaction with respect to an enzyme. Now, what do proteins do overall depends on their shape. So if a protein has a specific shape, it's going to have a specific job related to that. Well, if that shape changes even just a little bit, it might not be able to do its job correctly. So maybe it's supposed to squeeze things, but if I have a change in shape where it's locked up, it can't squeeze, then it can't do its job the right way. So this idea is called denaturing. So proteins are supposed to fold in a specific way to do a specific job. And if they unfold, usually due to high heat or acidity, then they'll kind of break down and now they can't do their jobs properly. So a great example of this is egg whites. Whenever you heat up an egg white, overall, you get that whitish color that turns rubbery. It goes from a liquid state to kind of like more of a rubbery state. And this is because egg whites are filled with proteins, and those proteins are denaturing or kind of spreading out. You can get the same effect done with an acid. All right. Speaking of acids, let's talk nucleic acids. This is basically your DNA or your RNA. So overall, this is your carbon, hydrogen, oxygen, nitrogen, and phosphorus components. You get a bunch of these together, you make nucleic acids, and these are called nucleotides, these single units. And these nucleotides are basically, depending on which sugar that you have here in the middle, deoxyribose versus ribose is whether you have DNA or RNA. And their jobs are basically to store genetic information, store information about the cells, how to make proteins, and the steps on making them. That's all for Unit 1, water, chemistry, and macromolecules. Hopefully I didn't talk too fast and stutter over too much, that you're able to actually gain some information from it. If you need to, please rewatch it, and have a great day.